So hopefully my voice is clearly audible to you all, and hopefully the slide that I'm sharing is also visible. So can somebody confirm?
Yes, yes, sir. It's visible.
Yeah, okay, that's fine. Yeah, so again, I welcome all of you to the week 6 live tutorial session for the course Bioinformatics Algorithms and Applications, instructed by Professor Michael Gromiha. And as always, I'm Sunanda Samanto, PhD scholar at IIT Indore, and I'll be taking you through some of the concepts and some of the example problems that Professor Gomiha has covered in the lecture series week 6 content, right? So we will start by looking into some of our earlier concepts, kind of just a brief review of what we had talked about earlier, and then we will try to delve into what Professor Gomiha has taught in this week's class. So can anybody quickly brief me on what are the topics that he covered in particularly in this week?
Sir, in this week, Professor Michael Green talked in detail about how to analyze protein structure in a 3D way, which means he also talked about major databases such as PyMol to know the further applications, and then he also described the factors how to visualize the 3D image in 2D graphs also. So it was majorly focused on 3D analysis of the protein.
Yeah. So protein analysis, as you know the structure, the 3D fold of the protein is particularly relevant to the functions. So there are various tools, as you know, for example, PyMol, Chimera X, Chimera, and so on, PMD, right? They allow you to see the 3D representations of each and every atom, as well as the backbone formation or the fold in different sort of interesting ways, like for example in ribbon structure, surface-based structure, something like that, right? And then they can also allow you to calculate various parameters. For example, if it's a deviation from some certain reference point or reference structure or distances, so on and so forth, right? So the 3D analysis is quite important. For that, of course, we will be requiring um, the the PDB files, right? Or any type of file that is uh, explorable or that can be visualized by these kind of softwares, right? Okay, so we'll try to look into that just in a bit. So let's try to cover briefly what we learned earlier, just in the earlier week's class. So firstly, we discussed about the non-redundancy of data points, right? So the non-redundancy basically um, is important for reducing the bias in a data. So as you know that if there are, for example, very similar data points that are accumulated in a data set, so that can skew the inference that is drawn from the data set in a particular manner, right? So that is important. It is important that we reduce the bias by introducing some sort of non-redundancy procedures, right? Maybe we set some sort of cutoff, and if we say that okay, if two data points have a similarity above this cutoff, then we'll only keep one of these data points, right? So very similar to that, we also discussed the one of the softwares, for example, CT heat, right? CT heat, and how does it reduce the data, the size of the database by introducing non-redundancy, right? By clustering all of the data points that are available in the data set, also using something called as greedy incremental clustering and something called the short word filter, the CD hit database can actually reduce the size of the database overall, right? The CD hit software. So here, for example, we had talked about earlier that the CD hit software uses something called short word filter, right? And it has certain limitations. For example, particularly if you are trying to characterize, or if you are trying to find the matches between the different data points that are available, for example, in this case, the different sequences. So in the worst case scenario, what can happen is that your mismatches are distributed very evenly. So that means that for certain type of certain percentage of matches, you cannot use all of the short word filters that are available. For example, here it is written that 80% for pentapeptide. Right? So you cannot use if the similarity is over 80%. So the worst case scenario, their mismatches will be evenly distributed, and you cannot use a pentapeptide or 5 short word filter, because in this particular subplot you see that you can never find a 5 word match between these two sequences, even though they are matching at 8%, right? So this is one of the softwares that we use, CD hit. Similarly, there are other softwares, for example, Blast Class, that Professor Promiha also mentioned. And then we just briefly went over the commands for the CD hit. For example, in our Linux system, we tried to use it, right? And using the sort of indication of parameter, for example, using minus C of 0.9, we can indicate that it is trying to match at 90% identity, or using minus N of 4, we can particularly specify the word size of 4 for the clustering, right? So particularly relevant to you, whichever software Professor Pumi has talked about, just try to remember a few input uh, commands kind of, or what parameters the software uses, so that this could be helpful in solving some of your assignment or exam problems. Okay, next we also learned about, since the relevance of clustering methods was discussed, right? So we also went over one of the very popular process or algorithms called k-means clustering, right? So anybody remembers what was the step-by-step process of clustering a 1D data, let's say for example this one, in the algorithms of k-means clustering?
Sir, it uses greedy algorithm for clustering purposes. It assigns if we wanna have 5 clusters, for example, so it assigns randomly the head of the cluster 1, 2, 3, and so on. Then it tries to formulate the shortest distance between those clusters and form those cluster, and it does in a repetitive manner, so that when it reaches a maximum point, then that it does not gain any more closer point, so it stops. But till then it keeps on going and going and try to find the closest distance between those between those points, random points, and form them into clusters.
Yeah. So it initializes the process by randomly assigning cluster centers, and then it allocates basically all of the other data points into to be part of the particular cluster, and then it computes the average point of the data within the cluster, and then proceeds to iterate through the process. And if it, for example, does not find a better overall deviation value, so that means the deviation value is not lowered at a subsequent step, so then that means that one iteration of the clustering is done. And then it starts the process again by again randomly assigning new cluster centers. And then through the iteration, if at the end of the process, the total variation within the data is reduced, so that means that the algorithm is still working, right? It can still find better outcomes. However, if the current iteration's total variation is not reduced from the previous iteration, so that means that the algorithm has converged, right? So that is kind of the basic steps of k-means clustering, you can say. We also tried to look into some of the examples where we tried to um, do like a 2D clustering via the k-means clustering algorithm of some data set, right? So that was one point. And then we also talked about the elbow method, where basically within cluster sum of squares value, at the particular point where this plot of the number of clusters versus the WCSS value is kind of flattened. So you cannot increase even if you increase the number of clusters further, you are not getting a reduced WCSS value. So that point is called the elbow point, and then that is the kind of the optimal number of clusters that we can go for while analyzing a particular data set, right? So that is something that we also talked over. We also went over the blast class and its commands, and then we then talked about basically the distances between different sequences, right? So the distances basically mean that how kind of far apart certain sequences are. It could be based on something like amino acid composition, as stated here. So here in this case, for Hamming distance calculation, we just had to do the composition calculation for particular amino acids in sequence 1 versus sequence 2, and then using the mode of comp 1 of I minus comp 2 of I, and then doing a summation of that, we could find out the Hamming distance of the different sequences, right? that have been provided. Okay. The same kind of process was seen in the sequence similarity analysis, and then we also learned about Euclidean distance, I think. So very similar formula, just to identify the difference in amino acid compositions for two different sequences, which can tell us how far apart or how close they are in their overall characteristics, right? Yeah. So we also did the Euclidean distance calculation. So again, pretty similar. Just here you do a square of the mod of comp 1 of I minus comp 2 of I, and then when you do a summation of all of that, at the end of the process, you do a square. Okay, that's the Euclidean distance. So what was the answer to this particular question? So which can represent a set of non-redundant sequences? So based on our discussion, we know that non-redundancy means that uh, in the data set, there should not be very similar data points, right? So that we do not introduce any sort of unwanted bias in the system, in the calculations. So anybody can try. Which one of the followings can give or can represent a non-redundant set of sequences? So I see C answer here. Yeah, so no two sequences should have a similarity of greater than 15%. Of course, if you go through the other options, first one is saying the no two sequences have a similarity of less than 75%. That's not the case, right? Then no two sequences should have a similarity of greater than 85%. So if two sequences have a similarity of greater than 80%, then that means that they are very similar, right? So that would mean that since here the statement is kind of negative, so that would not again mean a non-redundant set of sequences. For option C, no two sequences have a similarity of greater than 15%. So if the similarity is not greater than 15%, so that means that they are quite different sequences, right? So that would mean essentially that this is a non-redundant set of sequences, right? Okay. These kind of simple questions you can also get. So in the next question, I don't remember exactly whether we solved it or not, but here it is saying that what is the propensity for glycine to be in alpha helix in the following assignment. Now remember that when we wanted to calculate the propensity, we had to use this formula. Okay, so here first of all, uh, what you wanted to do is that the type of particular residue that we are talking about, for example, in this case, it is glycine, so it is an amino acid residue glycine, right? So firstly, we have to calculate that out of all of the glycines in the data set, how many of them are present in helix. Okay, so that is this particular formula, n of alpha of I divided by capital N of I. And then on the next step, we have to calculate out of all the residues that are present in the helix, in the whole data set, how many of them are present in helix. So firstly, you calculate for glycine, and then you calculate for all of the residues that are present in the given sequences, right? And then you just divide the first value by the second value, and then you should get the answer, right? So here in this particular sequence given, we have the assignment, the secondary structure assignment for each of the amino acids, right? For example, the alanine is a part of beta strand. Then glycine is also beta strand. Then your arginine also helical, right? Uh, E is also helical, Y is also helical, so on and so forth. Okay, so try to calculate it very shortly. We'll just see the answer. Okay. So first, try to calculate for all of the glycines that are present uh, in the data set, and then for all of the amino acids just as a whole that are present in the data set. Okay. So I see on answer A. Others can also try to do the calculation shortly. So first we have to find out how many of the type of the residue that we're talking about, for example, in this case glycine, are present in helix. Right? So here, let's say how many glycines are there in total. So glycine is your 1, 2, then 3, right? So out of 3, we can see one glycine is only present in helix. So uh, this particular value, this should be 1 by 3, right? As you see here. Okay? And then on the uh, whole residues part, so this particular value. So here what it should be that out of all of the residues, how many we have? 1, 2, 3, 4, 10 residues we have, right? So out of 10 residues, how many of them are total in helix? So how many helical secondary structure assignment we have got? So 1, 2, 3, 4, and this one is 5. This should be 5 by 10, right? 1 by 3 divided by 5 by 10. And overall, this should give you an answer of 0.67. So just quickly remember this formula because you might encounter it in several of the assignment problems. Okay. So yeah, so as stated in the chat also, answer should be 0.67 in this case, right? Now, um, there was one particular method that Professor Gomiha also talked about, where basically based on the propensity calculation, right? We can try to tell whether certain types of residues would be more prone to form certain type of secondary structures or would be more prone to disrupt that type of particular secondary structure, right? So based on this method called Chow-Fussman method, we can actually assign the different type of amino acid residues to be the certain secondary structure formers or breakers. Okay, so in this particular case, the question is asking uh, which of the following options are correct as per the Chow-Fussman method for alanine and tyrosine. So here we have the alpha C helix case as well as the beta strands case. So whether firstly alanine should be a former or breaker of alpha C helix and the tyrosine should be a former or breaker of the beta strand. So can somebody try to answer in the chat? We'll just see. Okay? So this is again based on the propensity calculations because so naturally, like if you find by analyzing a set of sequences, like in the case of different proteins, right, that whether uh, certain residues are prominent in a particular type of secondary structures, then you might also assume that that particular amino acid is a former of that particular type of secondary structure, right? Because otherwise, in nature, it would not be present in that like heavy amount in that particular type of secondary structure, right? On the other hand, again, on the opposite side, if you find out that you never like very rarely get to see certain type of amino acids in let's say beta sheet or beta strand, right? That means that that particular amino acid should be a beta strand breaker because if it's not present in the nature, then that means that that amino acid does not like to be a part of that type of secondary structure, right? So the Chow-Fussman method is kind of the same thing. Here, the answer kind of should have been A. Okay. Now, what is the Chow-Fussman kind of table? So here on this particular one, we are seeing that uh, all of the amino acids for alpha helix, beta strand, and turns, they have been assigned a particular value, right? So if you look for alanine, so the very first one, right? You see its alpha C helix related value is 1.42, right? Okay. Then if you look at tyrosine on the other hand, its beta strand related value, that is again 1.47. So if these values are greater than 1, based on the type of propensity calculation that is used here, so that means that that particular amino acid is present in very high amount in that particular type of secondary structures. So greater than 1 in this case means that that will be a former of that particular secondary structure, while on the other hand, if the amino acids, the chow phosphine value is less than 1 for a particular type of secondary structure, then that means that would be a breaker of this particular type of secondary structure. So if I ask, for example, from this table, so what would be lysine as a part of beta strand from this particular table? So should it be a former or breaker? So in this case, if you talk about lysine, of course, as you see, the value is 0.74 here. So 0.74, that means that it's less than 1. So it should be, from this particular assignment, it should be a beta strand breaker. So just remember this particular logic, and then you should be good to go for interpreting any uh, type of propensity from the Chow phosphate table. Okay. Okay. Now, so former and breaker, it's basically that from calculating composition through the naturally occurring proteins, when you see that certain type of amino acids are, let's say, very present in very high amount in alpha helical structures, right? So you will assume that, okay, so if this particular amino acid, I find it quite often in an alpha helical structure, so that means that for the stability of alpha helical structures, this particular amino acid is could be quite important, right? So that from that kind of way of thinking, we can say that this particular amino acid could be an alpha helix former because it stabilizes the alpha helix, let's say. That's why you see in nature that in proteins, large amount of that particular amino acid is occurring in alpha helical structures, right? On the other hand, if you rarely see one particular amino acid that it is not very often found in alpha helix, it is very rarely seen there. So then you will assume that, okay, if you introduce this amino acid in an alpha helical structure, so that will probably not be as stable as the natural kind of form, right? So then you will assume that from this way of thinking, you can say that, okay, this particular amino acid would destabilize alpha helix, and as such, we can call it a breaker. So that's the kind of way of thinking here. Whichever amino acid from a propensity calculation based on, let's say, naturally occurring proteins, right? Whichever amino acid we see in more higher amount in particular secondary structure, so that will be called a former. And if we see it in very less number of amount, very less number of quantity in particular amino acid, a particular secondary structure, so you can call it a breaker. That's the kind of logic here. Okay. So in this particular next question, mm, we want to uh, infer the, or we want to calculate the information content for a particular amino acid, which is alanine in helix. Okay. And now in this particular case, we have the GOR method, which is the Garnier Osguthorpe Robson method about calculating the information content. Okay. Now this particular method is using a formula that I have labeled here. So here, basically the information content as stated here is the left hand side of the equation here. Now, what are these different terms? Okay, so first of all, the SSI, this particular term, it means that the secondary structure at position I in the sequence. Okay, so that is what the SSI means. And then X is any secondary structure, which could be helix, strand, turns, coils, etc. And A could be any amino acid residue. Okay. So here you are saying that out of 100 alanine residues, 80 of them are in helix. Okay. So in this particular case, you see uh, you have to find out how many among all of the alanines that are present, how many of them are in helix and how many of them are not in helix. Okay. So this is what these two kind of notations are saying. So this is saying SSI, so that means secondary structure equals X, and then this pipe AA, so that would basically indicate that this particular uh, secondary structure and this particular amino acid residues coordination. Okay, and wherever you have this kind of curly symbols, it would mean kind of a not. Okay, so a negative um, kind of statement. So that means that the secondary structure, which is in this particular case we are talking about helix. So how many alanines are present in helix? So that would signify this particular one, and then how many alanines are not present in helix? So that would signify this particular one. And we have to calculate another value here, which is, now forget about alanine. Out of all of the amino acid residues that are present, how many of them are in helix, and then how many of them are not in helix? Okay, so that would give you, if you minus the second term from the first term, that should give you the answer. Okay, so remember, this is out of all of the particular, I mean, in this case, alanine. So out of all of the alanine, how many of them are present in helix? You have to calculate a probability of that. Okay, it's not just the simple count, but a probability. That probability, not probability, it's just, let's call it the fraction. Okay, and then out of all of the alanines that are present, how many of them are not in helix? Then out of all of the amino acids, how many of them are in helix, and then how many of them are not in helix? Okay, so would you try to calculate it out very quickly? So all of the information is given here. Out of 100 alanine, 80 of them are in helix, and out of 1000 residues, 600 of them are in helix. Okay. So let's try to calculate it out very quickly, and then we'll just see it. So as I said, if you just uh, use this particular formula, if you decode it, so out of, firstly you have to calculate for the particular amino acid, that is alanine, because you know the first part of the right hand side, it talks in the terms of this particular I, right? So I is in this particular, sorry, it talks about the particular um, formula in terms of the particular amino acid. That is why the AA is mentioned here. Okay, so out of 100 amino acids, we know 80 of them are in helix. So this particular first term here, this particular term should be 80 divided by 100. And then if 80 of them are in helix, then of course 20 of them are not in helix. So that means this particular term, so it will be 20 divided by 100, right? And now you just divide them. Of course, the 100 and 100 are getting cancelled out. And then you will get the first term basically here. And then out of 600, we know that out of 1000, we know that 600 overall amino acid residues are part of helix. That means this particular term here, it will be 600 by 1000. And then this particular term, since 600 are in helix and 400 are not in helix, right? So it will be 400 divided by 1000. So again, if you do the calculation, so it should be something like this. So 0.8 by 0.2 minus ln of 0.6 by 0.4. It should give you a total overall value of 0.98. Okay. So that's the formula here. First you calculate for the particular amino acid, and next you calculate for the overall all of the amino acids present. Okay, so just keep in mind this formula. And then here we have another question. which is asking which of the following amino acids has the least propensity to be a buried residue. Now, you can kind of find it out based on the characteristics of an amino acids. If you remember that we also earlier divided the amino acids in certain categories, right? Based on their hydrophobicity, polarity, and so on. So just very, shortly based on that, you can try to figure it out. So which of the following amino acids have the least propensity to be a buried residue? Just try to think, okay, so does that mean that the amino acid is more favored to be exposed to some sort of polar solvents, or is it the opposite? Is it hydrophobic residue? So on, right? So try to figure it out. We'll just see it in a bit. So just try to figure it out that whether a buried residue should be polar or non-polar. Yeah, I see can.
Sir, can you explain the calculation of the previous question?
So here basically what is it is saying that it is talking about the information content of particular amino acid in helix. Okay. I just discard all of this. The formula that this particular calculation uses is that uh, you have been given information for the overall amino acids in this particular protein. So here you have been given that out of 1,000 amino acid residues, 600 of them are in helix, right? So that means that of these 1,000 residues, 600 will be H, okay, and then 400 will be not H. It could be any other thing. It could be beta strands, turns, loops, so on and so forth, right? But we are just interested in helix or not helix. Okay. Then you have been given that for a particular residue which you want to be included in this calculation, so that is alanine here. So you have been given a total of 100 residues, alanine residues, that out of them 80 are present in helix. So that means that out of these 100 alanines, 80 ala, that is helix or part of helical secondary structure, and then 20 ala, that means this is part of some other secondary structure that is apart from helix, right? So based on just this, the formula that is mentioned here, that if you want to calculate the information content of alanine in helix, so you have to first calculate these two terms. Okay, so this is the first term. Let's say this is the second term. Okay. Now, what does the first term and second term basically mean? So they again are composed of two different parts, and basically inside this notation, what it is basically saying. So if you see the first terms, first part basically, so this particular one, so it is talking about out of all of the alanine that are present, because this is again saying in the terms of the particular amino acid. Okay, so out of all of the amino acids, out of all of the alanine that you have been given, how many of them are present in helix? Okay, so in this case, this should be 80 divided by 100, right? Out of 100 alanine, because 80 of them are in helix, so this fraction calculation, that would be 80 by 100. Okay, when you have this sort of curly, this curly symbol here, so that indicates a not, kind of like a negative statement. So this particular term, it is asking out of the 100 alanines that you have been given the information for, how many of them are not in helix? So since 80 of them are in helix, we know that 20 of them are not in helix. So the fraction calculation here would be 20 by 100, right? And then the second term that you have to calculate for. So in this case, again, you have a kind of a fraction value here. So this first calculation, this is basically asking the probability of uh, overall all of the amino acids, how many amino acids are part of helix. So this should be your 600 divided by 1000. And then this particular term that is given here, so it is asking that overall for all of the amino acids, how many of them are not helical secondary structure, right? That should be 400 divided by 1000. So that's it. Okay. So here in this case, you have to do ln of 80 divided by 100, again divided by 20 divided by 100, so 100 is cancelled out. You get basically ln of 4, right? And then minus you have the 600 divided by 100. Again, 1000 is cancelled out here, so ln of 600 divided by 400 kind of, right? So if you do the calculation, you would get a value of 0.9. So just that. For alanine, you have to calculate, and then for overall amino acids, you have to calculate. Okay.
Okay, so thank you, sir.
Hopefully this time it was a bit more clear. Yeah. So yeah, so we are talking about the propensity of certain amino acid residues to be buried or not. Now, in this particular case, as you know, which type of amino acid residue wants to be in a buried environment? Hydrophobic residues, right? But here the question is asking that which of them has the least propensity to be a buried residue. So the least propensity to be a buried residue, that should be some sort of polar residue, right? Polar residue or some sort of charged residue which wants to be exposed to a solvent kind of environment, right? And among all of these, we know that lysine is the one that is charged residue here, right? So that would have the least propensity to be a buried residue. That should be more kind of prominent in a surface type of position where it is more accessible to water. Okay, if you remember in one of our earlier classes, we also went over something called the hydropathy index, which indicated the free energy change of something, some amino acid, when we transfer it from a non-polar to a polar solvent, right? So in this particular case, we also saw that for the very negative valued hydropathic index amino acids, right? For example, lysine in this case, the free energy change of transferring that to a polar solvent, that is highly negative. So that means that this sort of change is very favorable, because we know that negative value indicates favorability in this case, right? So that is also one kind of correlation that you can have, you can draw. And then in this particular plot, we are also seeing that the surface propensity versus the buried propensity of all of the amino acids from the particular source that I have mentioned here. Okay. So in this particular plot also, you will see that the highly charged amino acids, for example, lysine, of course, its surface propensity is much, much higher as compared to its buried propensity. So it's not that there is no lysine ever in a buried position, but it's that that probability is very, very low. So a very less number of lysine will always be found in a buried position. Similarly, the case of arginine, aspartic acid, so on and so forth. Okay. Then for in the opposite side, we will also see that when the amino acid is hydrophobic in nature, you will find that their buried propensity is much, much higher than their surface propensity. So yeah, that's the sort of kind of method to find out these sort of answers. Okay. Yeah, we also talked about BLAST class in our earlier class, I guess. So BLAST class can take both protein and nucleic acid sequences, and it can be able to cluster those, right? And then we talked about CD hit. So do you remember what was the particular formula for calculating identity of protein sequences if we have a different length of sequences? Let's see, if we have two sequences that have different length or different size, so we divide the number of identical residues by the length of the shorter sequence. So similar in this case is not the correct one, because it should be the exact same residue for the CD hit algorithm, as I showed in one example in the earlier class. So the identical residue length divided by the length of the shorter sequence, that should be the precise answer in this particular one. Okay. So again, yeah, but yeah. Okay, so in this particular this week's class, so as Professor Gomiha went over the different sort of aspects of the 3D structure of proteins, right? So the 3D structure of proteins are of course very important, and one of the uh, methods that is often used for deriving the 3D structures is X-ray crystallography, right? X-ray using the X-ray diffraction, right? So this particular question that is asking, that is being asked here, it is asking the attenuation of X-ray scattering or coherent neutron scattering caused by thermal motion that can be described by which of these following factors or which of these following terms. And that now this particular term is quite important because it tells something about the behavior of certain atoms, kind of in this in a crystal structure. Okay, that's why this term is quite important. We'll also try to see where this particular term is can be found out in a crystal structure. If you just take example of one of the random structures in a PDB database, right? So it is also oftentimes mentioned there, and it can basically tell about the thermal motion or the flexibility or the fluctuation behavior of the particular atoms. Okay, so this term should be which one? I see answers in the chat. Others. So this is very important to know for categorizing different parts of a protein. You can say the behavior of certain atoms or certain regions even. And even when we are not just relying on the crystal structure, if you are again modeling the structure and then let's say we are going for something like simulation, then we can also find out the behavior of the particular atoms through this particular type of calculation, I'd say. So yes, as one answer mentioned in the chat, it should be B factor. Okay, so this is what B factor basically indicates. So if you go to the RCSB PTP website, let's say I'm just trying to uh, find out about certain structures. Okay, I'll just take one particular example. So in this case, we all know PTP database that stores the results of different crystallization experiments, right? For example, in this case, the method was used as X-ray crystallography or X-ray diffraction. And then the PDB database also gives us different informations regarding the resolutions, the R values, so on and so forth, right? As well as the associated literatures and whatever molecules that have been co-crystallized. So all of the data are present. But when we want to process the files, uh, the PDB files for certain calculations, we have to go for, of course, we have to first try to figure out how to download it. So you can go to the download files option and you can get it in the legacy PDB format or in any other formats. But we are just trying to here visualize clearly what this PDB database or the PDB files kind of contain. Okay. So if you just open up, of course, of the upper kind of header side, we will have a lot of different informations, remarks, and so on and so forth. But at certain point, you will see that the 3D coordinates and some of the other associated information are starting. Okay, so this is kind of the crux of the PDF file, and basically the most important part, which will be used in different calculations. And of course, when this file kind of ends, following that, we will find out that there are different connect information. So this talks about the different bonds that are present in between different atoms also. Okay. So in this particular case, if we just talk about, let's say, the 3D coordinate information and the other columns that are also present here. So can you say which of these columns or which of these kind of values are reflecting of the B factor for the specific atoms?
Sir, one with the sir, second last row.
Second last row, that means it's 55.67, 55.47, this one.
This one, yeah.
Yeah, exactly. So that is the particular column that is telling us about the B factor. Now, the B factor, of course, can be characterized or can be divided, let's say, different kind of ways. For example, as Professor Pomiha also mentioned that if the B factor is, let's say, around or less than 10, right, you can say that that particular atom is very rigid. Whether if it's like greater than 50, so that means that the particular atom is quite flexible. Okay. So in this particular case, of course, it makes sense that the atoms of the terminally positioned amino acid residues that will be highly flexible, right? So in this case, we see that 55.67, 55.47, 55.28, very high number of values exceeding 50, right? So that means that these atoms are quite very flexible. If you go further, you will see that the flexibility of the amino acid residues atoms, they reduce. For example, here we have 34. Then if you go in different parts of the protein, you might also see that the flexibility or B factor in the range of tens or twenties even, right? So that means those are relatively rigid atoms. Uh, so based on that, you can find out basically which region of the proteins are having a higher uh, kind of pattern of flexibility or pattern of fluctuation, and that which regions are also having a more kind of rigid type of structure with low flexibility or low fluctuation, right? So that is for each atom, you can try to figure out the B factor, and you can calculate that. And in the case of, for example, something like simulations, we have a very similar parameters called root mean square fluctuation, but it also can be taken into account by integrating multiple atoms for a particular type of residue. Okay, so those kinds of calculations can also be done. But since a large part of the class also was about the 3D structure, and the PDB is the main kind of format, so let's try to look into it. What exactly is there in this kind of coordinate columns? Okay. So first, of course, you have these columns, right? So 1, 2, 3, 4, 5, 6, 7, 8. This is just towards the right of something indicated as atom. So this atom indication usually means that this is a standard atom for a standard amino acid residue. Okay. Otherwise, if you see something different, for example, if a protein is co-crystallized with a ligand, then you will see that it is mentioned as a heteroatom or het atom. Okay. In this case, these are atoms. So these are the part of the amino acid residues. So what is the particular 1, 2, 3, 4, 5, 6, 7, 8, 9, 10? This particular column is saying the column 2.
Sir, it's representing the number of amino acid in a sequence.
No. So it's not representing the number of amino acids. It's actually representing the number of atoms. Okay. So if you notice towards the right, you will see that this is indicating the three-letter code of the amino acid. Okay. So this is valine. So the very terminal valine, or the very first valine that is captured in this particular protein. Okay. This part, this entire highlighted part is the first amino acid. Okay. And all of the atoms that this amino acid is consisting of, for example, it is having an N, which is the amines nitrogen. Then you have a Ca, which is called C alpha. Then you have CO of the carboxyl. And then you have the side chain atoms, for example, Cp, Cg1, and Cg2. So the numbering of these particular atoms, one after another, that is indicated by this particular 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, that particular column. Okay, so it is indicating atom number basically. The amino acid residues numbering, so that is indicated by this 363, 364, 35, 365, these particular column. Now, of course, here the numbering is not starting from 1, because in this particular protein, it's not just that the part that is crystallized, that is the whole protein. So there is also part before that, and there could be part after that also. So based on just that kind of sense, it is basically capturing the range that is starting from 363 up to the numbering that is given here. For example, it would be, I think, 639. Okay. So out of the entire length of the protein, the residue 363 to 639, that has been captured, or that has been crystallized in this particular PTB structure. So that is the residue numbering, basically. So this is starting from valine 363. And just towards the right of that, what is the value? Is 22.904, 21.687, this one.
Sir, x coordinate?
Yeah, so just after that, you have the x, y, and then z coordinate. So that is talking about the exact positioning of this particular atom in the 3D space. Okay. So this is the kind of the most important part, I guess you could say, because the coordinate information is what we want for any type of computational calculations, right? So that's very important. And just following that, you have this 1.00, 1.00. It seems that every value in this column is 1.00. So any idea what this one is talking about? 1.00. Let's say it could be something less than 1.00 also. For example, something like 0.95 or 0.9. So this particular column, it is talking about occupancy. What occupancy would essentially indicate is that, let's say for this particular nitrogen atom, the very first one, let's say two different positions have been captured out of all of the crystals that have been investigated here. I mean, in a single crystallographic experiment, right? So let's say two very prominent positions was captured, uh, and mostly, let's say out of the two, the first one was very high, uh, having a prominence. So that was like 0.95 or 95% prominence. And then the second one had very low prominence kind of, right? That has, let's say, 0.05 or 5% probability of occurring. So this particular value, if you just calculate it within one, right, the range of 0 to 1, so that would give you a value of 0.95 for the first position and then 0.05 for the second position. And then overall, if you combine this value, you should get a total of one value. So that means that the one value is indicating occupancy. And in this particular case, yes, the occupancy value is 1. So that means that very clearly this particular atom's coordinates positions have been defined. Okay. Even though it is having a very high flexibility, but we can say that this coordinate has been defined here, and it does not have any occupancy kind of classes. Okay. So that's what this occupancy column is kind of saying. And then we have the B factor values just towards the right of that, and then we have the simple indication of the atoms that are constituting of this particular uh, row, basically, right? So in this particular crystal structure, you see that this protein has been crystallized, and then on the bottom you will see that at some point there is a termination signal. Just that means that the one unit, so that is a separate unit, the protein that has been ended, or that is kind of ending there. And then the following that, you have the initiation of a het atom or heteroatom. So heteroatoms usually indicate something that is not the standard amino acid atoms. So in this particular case, this was a co-crystallized ligand molecule. And then just after that, you will see again, after the end of this particular co-crystallized heteroatom or hetero molecule, let's say, the ligand, you will see that the crystallized waters also, they also have their particular coordinates. And this crystallized water molecules can also be important in some cases, because let's say in a protein ligand complex, the water molecules are present in very close positioning to the binding pocket. So there could be some chance of forming a kind of like a hydrogen bonding bridge between the proteins and ligands via the water molecule. So again, when you try to process these sort of structures, you have to do it very, very sort of carefully. That's the kind of way I would say. Okay. So since we are talking about PDB structures, I guess we should briefly look at one of the examples, namely the example that I just showed you. So I'll be switching my screen. Just yeah. So hopefully this is visible now. The files, they are all text readable and then text editable files, because you can open them up using any kind of text editor that you have. You can also manually edit those files in the text editor, right? So in some sort of modifications, we also do tend to do that. But if you don't want to open it up using a text editor, you can also open it up using the particular dedicated softwares. For example, in this case, my default software in this particular system is UCSF Chimera. Okay. So what UCSF Chimera basically is, it is a 3D visualization tool as well as an analysis tool. Right? As Professor Pumiha has mentioned about the other kind of tools, for example, PyMol. So this is very similar very similar thing. So what can you do with this particular tool? Of course, you can get a full 3D view of the entire protein as well as the associated structures, for example, ligands and so on. You can change the visualization through different uh, available options that we have on the different tabs. You can do different types of calculations, of course. So let's just look at something like that. Okay. So here we will just first change the visualization a little bit. So if you go to the preset option, you can have a very nice depth cube rounded ribbon with white background as well as uh, let's say silhouette type of structure so that you each atom and each kind of ribbon's outline is very clearly highlighted. Okay. Now in this particular structure, you see that certain regions have uh, not a specific uh, kind of 3D structure that is given. It seems that this particular case, the associated 3D structure is missing. So these are your missing regions that was not quite captured by the X-ray crystallographic process. And if you go to the sequence, you will see that the areas that are highlighted within this red sort of rectangular boxes, so that indicates your missing residues. Okay. So of course, before processing the PDB structure for any type of calculations, you first need to model this particular residues. Now, in this particular tutorial, we will not look at how to do that. You need different types of software, different types of tools. For example, modeler that can be associated with usage of Chimera also. So you can use that to build your model. The process is kind of the same when we learn about homology modeling, about how to uh, do sort of build up protein model based on other homologous sequences. We will also find a very similar sort of application of the loop modeling there also. Okay. So anyway, so using some sort of tools, for example, it could be the freely available UCS chimeras associated a modular structure uh, from Sally Lab, or then you could have some sort of proprietary softwares like Schrodinger. They also have their different loop modeling tools. But after you are done modeling, you might want to look into the structure a bit. So let's say you are interested in changing the visualization. So you can go to the action option, and then you can go to the color, you can go to the all option section, and then based on your preference, you can just color different sort of secondary structures or different selections in different ways. For example, I am just doing light gray, basically gray or dark gray color for the protein part, for the ribbon part particularly, right? Uh, then if you notice on this particular section, you have a residue or a particular sort of molecule that is labeled as 585. So if you remember this particular PDE file there, if you go to the display file again, okay, not this one. Yeah, if you go to the legacy PDE formats text file, we'll notice that on the very bottom, after the protein information ended, you saw that there was something called the head atom, right? As we talked about earlier. And this head atom's residue name is indicated as 585. So if you do the if you correlate that with the 3D visualization, you will see that this 585 is this particular one. Okay. So this is a co-crystallized inhibitor. It is a kinase molecule and it is an ATP competitive inhibitor for that. Let's try to change the visualization of this also. So you can do it in the action tab. So you can select atoms and bonds. You can color it in different color. You can color it by heteroatoms also. Um, then you can also highlight the atoms and bonds in different representations as well. So kind of like this. And let's say you want to highlight or we want to just hide the binding pocket residue side chains. You can also do that. You can just select, for example, the structure and you can select the protein. If you do the actions, atom slash bond, and then hide the options, so it will hide all of these three kind of side chains that have been visible in the structure earlier. Okay, it will keep the ribbon intact, but it will hide the atoms and bonds selection for this particular case, right? You will also notice that with the clear selection option here, you notice that uh, there are different water molecules that are present that are very close to the binding site. So this particular water molecules, they are in default, they are visualized the usage of chimera. That does not mean that there is not any other water molecule in this particular protein complex that have been crystallized. Okay. If you just go to the select option can help you in selecting a variety of uh, particular kind of structures, atoms, so on and so forth. For example, here if you can go to the residue section, you can select any type of standard amino acids, you can select any type of non-standard amino acids, for example, water, ligand, so on and so forth, right? You can select by amino acid categories also. But in this case, let's say we select water and then we go to actions, atoms, bond, and so you see that a lot of water molecules have been present that are in this particular PDB structure. Mainly, when you want to analyze the structure, just be careful about whichever amino acids are, whichever water molecules are very close to the binding site, because that can be facilitating some sort of interactions. You don't know that beforehand, so you might have to do a lot of literature survey and so on and so forth for that. Okay, if we just hide, if we just do clear selection, first hide this particular selection. Let's say you want to know, okay, this seems to be the protein structure. So the ligand is supposed to bind to a cavity, right? So what is the surface view of this particular protein? So that would allow us to see, okay, where in which particular cavity this particular ligand is binding. So if you select the entire protein here, what you can do is that you can go to the surface option under the action tab and you can just press show, and then it will show you a 3D surface representation of this particular protein. And then you will see that in this particular cavity. So this is kind of the ATP binding cavity where this ATP competitive inhibitor is being targeted towards. So it is binding in this particular cavity. So that can give you a visual sort of aspect of, okay, this is where my particular ligand is binding, right? So as you see, by analyzing through the efficient chimera, you can get to not only look at the particular proteins and the complexes quite thoroughly, but you can also create, good quality publication, quality images from it. There is options for that as well. You can go to file, you can go to save image, and then through the options that have been available here, image width, image height, so basically kind of a way of increasing the resolution kind of in your image. You can save a very high quality uh, publication quality kind of figures from this particular software tool also. So let's just hide the surface here. We go to actions, we go to surface, we go hide, right? That was the original ribbon presentation. Uh, so not only that, there are different other calculations that you are actually able to do throughout the use of USS Primena. If you go to command line, there is also command line bar. But let's say I want to calculate certain distances, right? So let's say I just clear the selection here. And let's say for some reason, I am interested in calculating distance of particular amino acid residues. Okay. So here we have a glutamic acid from the alpha helix. If you select this one and if you just go to actions atoms less one show, the side chain orientation is also becoming visible here. Okay. Now, as it happens that this particular type of proteins, they have a very important salt bridge interaction. Okay, so this is very important in kind of assessing the stability of the region close to the binding site for this protein, and quite important in determining its active state also. Okay, now let's say you are wanting to find out, okay, in this particular crystal structure or in any structure that we might get from something like a molecular dynamics simulation, whether this protein is staying active or not. Let's say one parameter that you can calculate here would be the distance between these two particular uh, amino acids. So this is quite well established, right? Now, how do you calculate distances? Of course, to calculate distances, you have to go through the formula for distance calculation using the x, y, and z coordinate, right? So that is one way. But of course, since we have the use of the software here, we can easily do that. Okay. So for this calculation, let's say we are selecting two different atoms which are standard way they are selected in general. So let's say I want this particular CD atom. Okay, of this particular glutamic acid 420. So these two, remember, these two are very important for the active state determination of the protein. If there is a salt bridge present, so that means that the particular protein state is active. Okay, this particular kinase enzyme. So here GLU 420 we can select the CT, and then here again for the lysine 402 we can select the NZ, the nitrogen atom, by just pressing Control Shift on my keyboard, and by selecting it, we are seeing that these two atoms have been selected. Now, if you go to the tools section, we'll see that under the tools section, a variety of different calculations have been indicated here. Okay, one of the options in this particular case, if you go to the structure analysis part, you will see that there is also an option for calculating distances. It says that distances, right? If you click this one, with your selected ID atom 1 and ID atom 2, if you just create, then you will see that one particular distance has been created here with the value of 3.799 angstrom. Now here it is colored in yellow. Of course, you can change the color as per your convenience as to what looks good in this particular sort of white environment, white background. So you will see that the distance is also indicated here as 3.799 angstrom. Okay. So since within 4 angstrom we can consider it as a salt bridge interaction. So that means these two amino acids, the lysine and the glutamic acids, they are having a salt bridge interaction in this particular structure, we can see. Okay. So apart from distance calculation, of course, you can do a variety of analysis, as I said, in this tools tab. You can go to structure analysis. You can find hydrogen bonds, distance classes, distances, angles, torsions, so on and so forth. Then you can also compare different structures using the built-in matchmaker tool. You can align different structures. You can do ensemble clustering, so on and so forth. Then you can also do surface or binding analysis. Here you will find a lot of different options. For example, finding H bond, hydrogen bond, electrostatic surface coloring. You have the view dog, dog pip, autodog FINA. These are also integrated part with other softwares that are available, other tools that are available for different other sort of analysis. You can see, for example, docking. We do the dog play, autodog FINA. These are for molecular docking, right? Which will also talk about some of the other classes. You can edit structures, of course. You can model defined loops. I was saying that you need you need to integrate the modeler software from a different group, and you can integrate that through uses a primer for modeling or defining loops. You can do structural minimization. Minimization is basically a way to reduce the static classes that is occurring within the structure. That can give you basically more well optimized structure to start certain calculations. Okay, so you can do that, those kind of calculations also. You can also go for the molecular dynamics analysis. If you have a calculated file, you can visualize it through uses a primary in the form of a movie or a slider, where upon selecting the entire protein or doing some side chain visualization or protein ligand complex kind of formation in any kind of way you do the visualization, you can see how this particular pattern is changing throughout the kind of trajectory time scale. Okay, so that will give you an idea about what is changing or what is kind of being modified at the structural level throughout your simulation trajectory. So that is also quite helpful. You can do the structural analysis, higher order structure calculation, so on and so forth. So the point is that this type of visualization tools are quite important, and then they will allow you not only to do the calculations, but they will also allow you to look into your data, whatever you have, the trajectory you have, in a more thorough way. They will help you to produce good publication quality images as well. And a variety of options are there, of which you need to select what you want to do basically from all of these options. And basically, you need to play around all of the options to be able to understand the entire breadth of this kind of software. Okay. So that's just very shortly something about UCSF Chimera. So I'll just go over something else very quickly. Yeah, please. I see one hand.
Sir, can we use Pymol in the same way that we use Chimera?
Yes, definitely. Pymol is also
Okay.
a very same type of
Sir,
software. Yeah. Mhm.
Sir, actually, I downloaded PyMol as suggested by the Professor Gromio, Michael Gromio also, as he mentioned PyMol in it. So I kind of visualized some basic proteins like hemoglobin and hemocyanin also. It was very interesting.
Yeah. So PyMol is also very similar, where you will also have a lot of different functionalities alongside the visualization aspect of it. Uh, Other kind of tools are UCSF Chimera X, which is the same group of UCSF Chimera, so the same research group that is developing it. And then you also have VMD. So VMD is also another quite popular tool for visualization and calculation and so on and so forth. So you just need to get familiar with one or two of these tools, and then that will help you throughout your analysis basically. Okay. So they're quite similar software. Some commands and some options, etc., will be different, but overall they do the same thing. Okay. So here, for example, I want to find out. Okay. So okay, first let me clear the distance that has been created. If you go to the tool section, structured analysis distances, you can also remove the distance that have been created here. So it's 3.79 and highlight. Just want to remove it. Okay. Let's close it. Select structure protein, and then we just want to find uh, all of the atoms apart from the ligand. Okay. Okay. Now let's say we want to find out, okay, which particular amino acids are uh, corresponding to the binding site, or which particular amino acids occupy the binding site in this particular protein ligand complex. Of course, you see that the ligand is staying here. What you can do is that you can manually go through the surrounding amino acids, for example, serine 379, glycine 378, so on and so forth. These will be very close to the amino acid, right? So they could be part of the amino acids, forming some sort of interactions. It could be hydrophobic interactions or some sort of hydrogen bonding, something like that, right? But one way to uh, kind of uh, very easily define the surrounding area of a ligand in this protein is that, let's say uh, we take this ligand as a reference point, uh, just the center of mass of the ligand as a reference point, and let's say we calculate what could be the amino acid residues that are present surrounding this ligand that are within a 5 angstrom or within a certain angstrom cutoff range. Okay. So if it is very close to the ligand, of course, that could be part of the binding site, right? So we can do the very same thing here. So if we can select the ligand, for example, you can go through the select option, you can go to the structure, you can select the ligand in this way, right? You can also just press control and click on one part of the one atom of the ligand. You can press the upwards key and it will select the entire ligand in the same way. What we can do now, we can go to the select button and then we can select, or we can go for the zone selection. So the zone selection tells us different options. So the first one is telling that out of the selected atoms and bonds, of all of the selected atoms and bonds, we are taking as a reference point. So it will first kind of calculate its mean position, kind of the center of mass, and then it will say that the angstroms, it will select basically whichever uh, amino acid residues, or all atoms and bonds in this case, that are at 5 angstroms from the currently selected atom. So that is what it will select. Okay. So if you click OK here, and then it selects the different amino acid residues, and then if you go to visualize and then see the sequence, then you will see that different amino acid residues are being highlighted, right? So since the different amino acid residues are highlighted, they are coming within uh, the 5 angstrom range from this particular ligand. You can quickly say that, okay, so this particular amino acid residues could be part of the binding pocket or involved in some way, because they are staying in the close vicinity of this particular ligand, right? So that is one quick way to define the binding pocket of the ligands. Okay. So if you do the clear selection option here, you can go to the actions options and then atom slash bond will show, and then you will see that the side chains are also visible. Uh, sorry, hide the ligand here. you can go to the tools and then you can find out, for example, different hydrogen bonding patterns here also. Let's say select the ligand here, you go to tools, you go to structure analysis, you go to find H bonds. Uh, and let's say we do both intermodal, intramodal. We go to okay. So in this particular case, what I did is that I did all of the intramodal and intermodal hydrogen bond calculation. So basically, within proteins as well as the protein ligand hydrogen bond formation is kind of shown here. So here, if you go to particularly for the protein ligand calculations, you will see that this particular nitrogen atom is forming a hydrogen bond with the amino acid residue GLU452, and then this particular nitrogen atom of the ligand is forming hydrogen bond with alanine 451. So you can also try to visualize this. This is the backbone kind of hydrogen bond formation, but that's fine. So you can do the visualization in different ways, and then you can see the different type of bonding patterns in this case. Okay. So anyway, so that is basically the point of the different options and the different calculations that are present in UCSF Chimera. You can play around UCSF Chimera or any other type of tools, this type of tools like PyMol, VMD, so on and so forth in your, in different times. Then you will see how to do the different type of calculations as well. Okay. So I guess that was quite briefly it. Yeah. Now, in this particular case, this example, again, this question is again asking about or trying to infer about the example from B factors, right? So here we are saying that the temperature factor, which is basically B factor, of less than 20. If it's less than 20, then what should this particular atom be? Should it be rigid, flexible, located in fixed position, or none of the above? So yeah, so option A, I think I see in the chat. So it should be rigid, right? Because less than 20, that means that the P factor is quite low. Its thermal fluctuations caused by thermal motions, that is also quite low. And that means that it is essentially a rigid amino acid residue. If it is higher value like more than 50, then it would be flexible amino acid residue, or let's say around 45, 50, that would also be kind of classified as flexible. Uh, the atoms are not located in a fixed position because all of the atoms they have a certain degree of motions. Okay, the degree of motion is more for certain atoms. Depends on the positioning and the buriedness and the different kinds of interactions that they are forming, so on and so forth. But they are not always located in the fixed position. They have certain degree of motion at the very least. Okay, so in this case, the answer would be rigid. So we saw this one earlier. Yeah. So again, since we are talking about the structural kind of aspects of proteins, uh, let's say uh, the same protein, the same residue length that have been crystallized by different research groups, right? Of course, there would be differences in the resolution, uh, but there is one statistical parameter that can say say us about or give you information to us about what is the difference between these two particular structures for the same protein. So there is one particular statistical parameter mentioning that, which is also quite highly important when you talk about analyzing a long molecular dynamics trajectory analysis. Okay, so what happens in molecular dynamics simulation, as we will know in the later kind of classes, is that so it is basically integrating the position of the different atoms throughout time, right? It is integrating at a particular time step, and how the structure of the molecule or the proteins and ligands and so on and so forth are defined. They are defined by something called force fields. Okay, the force fields is basically having information regarding the potential of the different terms that can constitute the particular molecule. For example, you have bonded terms, you have non-bonded terms. Um, the non-bonded terms will, consist of your van der Waals interactions, electrostatic interactions that can tell us about the local and the distal environment. And then there is the bonded terms, how two atoms are connected, so which angle they are forming, what is the dihedral between four atoms, so on and so forth. So all of these and the related potentials, they are embedded in force field, and that is used for calculating, let's say, new position for all of the atoms after a certain time interval or a time step, let's say. Okay. So if you do the calculation again and again and again, you will have a continuous trajectory for one particular um, system. So if the system could be your biomacromolecule, something like protein ligand complex, something like a nucleic acid, anything, right? But let's say you have now information about the behavior of a certain system throughout time, certain time, let's say nanosecond time scale or microsecond time scale, something like that. How do you know from the initial structure how much the new structures in the trajectory that is deviated? Okay, so this deviation parameter will tell us, as compared to your initial structure or any kind of reference structure, what is the difference of the newly obtained structures in your trajectory or newly obtained conformations in your trajectory. So one such statistical parameter, I think Professor Komiha also mentioned this, that is called root mean square deviation. So root mean square deviation or RMSD, you will see it all the time in all of the computational studies, because it is kind of a necessary parameter uh, to assess structural stability, to assess convergence of simulations, to assess conformational dynamics, all of that. Okay, so it's very important. And what it basically means is that in this particular case, if we talk about mass weighted RMSD, so the formula that uses is something like this. So here xi is indicating coordinate vector of target atom I. Let's say we are talking about just the first atom, and then yi is indicating the coordinate vector of the reference atom I. So the target atom, that means that, let's say in a trajectory, the particular nitrogen of alanine, uh, that has now a new coordinate, right, after your simulation. But in the initial structure where you started your simulation from, that had a different coordinate, right? So that is the reference coordinate. And the target atom is basically saying the new coordinate kind of, okay, after simulation, let's say in that. Okay. So uh, the root mean square deviation, you can see it through chimera also. For example, in this particular case, I just to try to show you how to visualize structures using chimera. Here we are trying to open up four different structures, four or five different structures in uses of chimera all at once. And what these structures basically represent, so they are kind of the simulated trajectories part. So this gray one, not kind of gray, so this particular structure, which is indicated as structure 0, kind of gray kind of structure, right? So this is your initial structure, and the other ones here that are kind of or rest up together. Uh, these are your trajectory kind of structure that you get for your simulated proteins. Okay, so through the option of usage of chimera of structure comparison, you can find out something called the match method. Okay, so now what this match method is basically doing, it is trying to superimpose one particular structure to something called a reference. Okay, so if you take the initial structure or hashtag 0 as your reference, and if you say take, let's say, structure 1 as your structure to match, if you click on OK, you will see that the blue structure is now overlapped onto the gray structure, which will give you kind of the superimposed pose. You will be able to visualize where change has occurred. For example, in this particular loop region, you see a lot of changes. And alongside that, if you open up the reply log, we'll also see the RMSD value um, of the whole protein, and the structures that you are trying to match, the RMSD difference between them have also been indicated. So here it is saying that between 2 to 2 atom pairs, it is 0.976. Across all 277 pairs, that is 3.162. So this is one part that you can see. If you have just few structures, you can able to visualize them in terms of the structural superposition also, as well as in terms of the values. So if I just quickly superimpose all of the other ones as well. You go to the match to that quickly. If you want to see and then 4 also, you press OK. You'll see very quickly that all of the structures have been superimposed on this particular initial structure. Okay, you can visualize the differences, their similarities, in which regions change have been identified. You can also go to the reply log now, and then you can see that the different RMSPs after each superimposition, they have also been indicated in this particular case. But that is only for a handful of structures that you have, right? If you are talking about a simulation trajectory, what happens is that simulation trajectory is very kind of large scale analysis where you don't only have a couple of or a handful of structures to investigate, you have a whole array of uh, confirmations to analyze. Okay, so basically in this particular case, after each time your snapshot is being saved, it could be certain picosecond time scale, uh, let's say 10 or 20 picoseconds. You will see that if you are uh, saving your confirmations at that frequency, in your total overall trajectory, a lot of data or a lot of structural snapshots will be generated. And if you do a calculation or large scale calculation of any type of RMST profile, you have to plot it based on the time evolution. Of course, the time evolution means that you will be able to see in a stepwise manner or in a time-wise manner how your protein is changing, which corresponding to something known as the difference, right? For example, in this case, in this RMST file, we see that in total, if you go to the very bottom of this particular file data set, you will see a total of 20,000 snapshots have been saved. So that is the left-hand side column. Corresponding to that, on the right-hand side, you will see that the different RMST values have been indicated with respect to the reference. In this case, the reference was the initial structure. That means that how much the protein is now deviating as corresponding to our reference, and we are getting a profile of that throughout the entire time. Okay, throughout the time that our simulation is running, which in this case is 20,000 simulations, which generated not 20,000 simulations, 20,000 snapshots, which were generated throughout the course of 200 nanosecond simulation time scale. What you can do is you can use some plotting tools, and you can see the time evolution of this particular parameter or RMST throughout all of the snapshots that have been saved. So this x-axis, they are taking about all of the frames or snapshots. The y-axis is your RMST value. And then you will see that, of course, it does not start exactly as zero value, because the part that we are using for the calculation, that is a production trajectory, production non trajectory. Before this, there also is some sort of equilibration phase. Before that, there have been a minimization kind of process. After that, we have also heated the conformation up to a 300 Kelvin temperature. Then after that, the equilibration, and after that, the production run has started. So before the production run, a lot of structural deviations have already occurred. And now, at the start of the production run, it is starting at a 1.5, around 1.5 to 2 angstrom RMST range. And then you will see that as the simulation is progressing, we are seeing in the initial state that there is some change, some slight fluctuations occurring in this particular range, the 4,000 to 6,000 snapshots. That means the structure has yet not become a quite stable one, right? After that, you will see the structure did start to get stabilized, and towards the end in this particular region, after let's say 14,000 to 20,000 snapshots, it's quite flattened out. The RMST profile is quite flat now. It's kind of ranging around like 2.3 to 2.4 angstrom sort of RMST range with respect to the difference. And that means, so what this particular type of plot will tell us is that if the deviation value is now flattened out, if the deviation value is not changing that much, so that means that the system has stabilized, or one other way to say it is that the simulation has converged. Okay. So basically what I mean is that the RMST profile is quite important for uh, analyzing the structural stability and the simulation convergence of your particular system. So I believe that the RMST profile was discussed by Professor Bromina, so that's why I went over it a bit. But anyway, so let's get back to the slide. Yeah. So this particular parameter, as we know, was called RMSD. And then the next particular question, again, it is talking about the 3D structure of the protein, but now in terms of solvent accessibility. So it is saying that the solvent accessibility is of particular residues that have been mentioned here in terms of, let's say, angstrom square values, right? So it is an area value. It is a kind of very similar to the solvent accessible surface area value that have been given as 0.2, 52, 1.2, and 25. So what would be the residues located at the interior portion of this protein? So in the interior portion is meaning in the sense that the residues are buried, okay? Buried in nature versus solvent exposed or surface located nature, okay? So just try to think of it in a way that um, if the residues are to be in interior position versus in a surface exposed position, whether the solvent accessibility will be high or the solvent accessibility will be low. Okay. So since we know that the residues that are outside the surface, outside of the protein or in a surface location, they will have more exposure to the surrounding water, right? So that means that the higher sort of value residues, for example, higher solvent accessibility having residues, right? For example, 50 to 25, 75. So that means your GLU 8, your aspartate 10, and alanine 11. So they are not part of the interior residues. They are located at the exterior or at the surface exposed position because they have a higher solvent accessible surface area. On the other hand, you will see that the alanine 7 and valine 9, they have the solvent accessibility values of 0.2 and 1.2. So this particularly means that alanine 7 and valine 9, due to having very low solvent accessibility values, they should be located at the interior position in this case. Okay, so that's how you just figure out this sort of questions. To see what other concepts there was. Okay, yeah. So there are some concepts regarding contact maps and then the resolution of PDB structures, so on and so forth. But I guess I will discuss those in the next class. Okay. Yeah. So for today, I think I'll just wrap up the class. And then in the next.
Thank you, sir. Sir, can you?
Yeah. Yeah.
Thank you so much, sir. Before wrapping up, can you explain the previous question?
Which one?
Just yes, this one.
Okay. Yeah. So in this particular case, it is asking about the solvent accessibility. Okay. Now, the solvent accessibility means that whether a certain residue is more exposed to the surrounding water or whether it is not exposed to the surrounding water. If it is more exposed to the surrounding water or surrounding solvent, that means that it is located in an exterior position. It is not a buried residue, right? It is located at a surface kind of position. On the other hand, if it is less exposed to water, if it is solvent accessibility is low, so that means that this is a buried residue. So in this particular case, residues located at interior, what the interior term basically means, it means that it is asking about buried residues, okay? And buried residues, since those residues will be in the inside part of the protein, basically away from the solvent or away from the surrounding water. So that means that they will have a very low solvent accessibility score. So in this case, the solvent accessibility score is mentioned in Armstrong square. So I think it should be something like solvent accessible surface area. So the lowest solvent accessible surface area molecules, they are the part of buried kind of environment, because the more solvent accessible
Yes, sir.
exposed there. Yeah. So that is the kind of logic here basically.
Okay, sir.
Hmm. And
Okay, sir. Thank you so much, sir.
yeah, solvent accessible surface area is also quite an kind of important concept, because you will see a change in this type of parameters in various kind of scenarios. For example, let's say in your protein structure, let's say a loop structure, like a loop structure was oriented inwards in a buried kind of position versus it is oriented outwards in a more exposed type position. So the solvent accessibility of this loop region will be changing. Right, and from that you can infer that okay, some sort of structural readjustment is going on in this particular region. It could be very relevant to your functionality as well. For example, in the case of kinases, so there is a particular segment, particular region called the activation loop, and the activation loop it is kind of basically the part where your substrate is binding. Okay, so if it is orientated inwards, meaning having much lesser solvent exposure or much lesser solvent accessible surface area. So it is blocking the substrate binding pocket. On the other hand, if it is orientating outwards in a more solvent exposed position, so that means that the substrate binding pocket is free basically. That is nearby the activation loop. So based on the calculation of Sasha, you can infer the structural readjustment or structural rearrangements, and then you can see whether it has any functional sort of relevance or not. For example, in the case of activation loop movements inwards and outwards. So this type of calculations is also quite important. And a large scale change for the overall solvent accessibility of the protein. If you see any sort of change, then you can also assume that there are some sort of large scale changes in the structural or conformation that is going on. Okay. So that is also one thing. If your ligand is, for example, binding to your binding pocket, then of course it will displace a lot of water. That means the binding pocket solvent accessibility will also reduce. So in this sort of way, from solvent accessibility also you can infer different type of uh, you know patterns in your particular system. So those are also quite important parameters to take note of and to calculate and to analyze. Okay. So yeah. So I guess I will just wrap up today's class then. Thank you so much, all of you, for joining. So there will also be one TA session tomorrow. Please also try to join that. Also, please try to go over the Professor Promiha's lectures in detail, because those will be what you will encounter in your classes or in your exams and assignments. Okay. So thank you, everyone, and we will meet in the next class. Is there any study material apart from the PPT? So apart from the PPT that we share, just go over Professor Gomiha's particular slides. I guess from the uploaded lecture videos, you can take screenshots for those particular slides. There are other resources also. For example, in the bioinformatics, for the bioinformatics course, I often take references from a few books, which I have also included in the Google Drive folder. So if you find certain parts, let's say, confusing, or if you are interested to know more about certain things, you can also refer to those books. Or there are different blog posts and the different websites, so on and so forth, that can also give you information regarding particular terms. Okay, so you can look it up online as well. You can go through the books that I also included in the Google Drive folder for your further knowledge also. But mainly, I would say from Professor Gumiha's slides, the concepts that he is particularly talking about, just focus more on those, because those are what you will encounter in your exams and assignments. Okay, the others are for your practical skill building, something like in that sort of direction. But for exams and questions and answers, focus on those particular slides and of questions of similar pattern to what you see in your assignments. Okay, for your final exam. Okay, yeah,
Okay, sir. Thank you, sir.
yeah. Thank you so much.